Welcome to the Biomedical Research Lecture Series for public and patients. My name is Sallie Lamb. I'm Professor of Rehabilitation here at the University of Oxford, and I'm the founding director of the Oxford Clinical Trials Research Unit, which is one of the bigger clinical trials units in Oxford. I'm a lead investigator for a number of large clinical trials. I'm a physiotherapist by background. I'm going to pose and discuss a question about whether or not clinical trials are the most important medical invention to date. I'll cover a number of topics that are of importance to understanding the science and philosophy of clinical trials. So I'm going to talk a little bit about the history. I'm going to talk about important concepts, and they will be randomisation. I think that's the most important concept that I'd like to get through this evening. Placebo, masking or blinding, which sounds horrific, but actually is intended for a good purpose, and some discussion about statistics as well. I'll talk a little bit about the law. I'll talk a bit about regulation and good practice. And I'm going to also show you some of the issues that are involved in running clinical trials and what it actually means for us back in the office, and hopefully for you guys out there who might be asked to participate in clinical trials as well. A clinical trial is in many ways kind of like the last frontier of what I call medical discovery. It's where we actually take new things, be it a medicine, a surgical procedure, a new therapy procedure, and we test them in humans. So they've come out of the lab, or they've come out of some kind of conceptual brainstorming, and this is the first time we put them into man. And then we like to kind of discover even more, and we'll do a whole series of different types of clinical trials to try and evaluate, does it work? And then also, how much might it cost if it looks like it works? Is it worth the taxpayer paying for it? And these are really important questions for our country and our economy. Clinical trials are also where fame and fortune and careers can be made. And I think that probably a number of you will be aware of the work of Ben Goldacre, who has really substantially raised the profile of bad science, as he likes to call it, and what I call it is misconduct in clinical trials. It is obviously important, and I'm going to talk about that a little bit more, but I'm going to talk about human flaws. And they're flaws that we all have. We have them as trialists, we have them as individuals in society, and we need to understand them so that we can design good, robust science. So for me, clinical trials are a voyage of discovery. They involve a massive enterprise of human endeavour, and that's from patients, clinicians, scientists. They require teamwork and creativity. So just so we're all starting from the same page, what is a clinical trial? It's an experiment. It tests a clinical intervention in a patient population, and it should be a fair and unbiased appraisal. And that's really important because these are experiments that we base clinical decision-making on and that we base the commissioning of health services on. So fundamentally important that they are conducted properly. They inform patients, public, families in their decision-making. They are an essential step in the regulatory approval of medicines and medical devices now. So you cannot have a medicine approved without having gone through this process. And it tells us not only about the benefits of a treatment, but about the harms as well. So I've already discussed costs, but benefits and harms is something that we capture. As I've said, we do trials for lots of different reasons. And this is just to give you a sense of the different types of trials that we run. And this is a slide that I showed to lots of trialists across the country, and I say to them it's very important to understand who the trial is for and what you are doing it for, and people can get themselves in a little bit of a mess with this. So I'm going to show you actually a visual which is a little bit easier to understand. We have proof of concept studies and early phase trials, and these are really quite small trials, usually of short duration. And I think you've probably heard of the Middlesex trial problems that happened now probably about 5 to 10 years ago. These are the early phase trials where we really are kind of putting something into people in very small number and being very careful to make sure that we kind of completely understand the effects in particular of medicines. And then we move to late phase trials, and then finally to implementation and impact. And you might hear these referred to as phase 1, phase 2, the effectiveness trials are phase 3 trials, and then phase 4 implementation and impact. They just kind of give you this sense of moving forwards. So let's have a little look at some history. This is James Lind, who was a naval officer in 1753. And he's actually the person whom we attribute the first record of a clinical trial to. He's probably not the first person who did a clinical trial. We know that the Greeks liked to experiment. I know this from my own area of clinical practice. They liked electricity, and they used to get electricity out of stingrays. So I do know that from my kind of formative years as a physiotherapist. But this chap is the first one to have actually written down what he did. So it's truly an experiment. He's done something and he's reported it. And this is what he did. He took 12 patients. So he did one of the very important things that we have to do in clinical trials, which is to have an estimate of the number of people we need to put into a clinical trial, and that's something I'll come back to. He knew what he was studying, which was scurvy. Unfortunately for him, he was at sea and he knew where he was. What he did then was he divided these patients up. So he knew that he had to compare between patients in order to be able to see which was the most effective of these treatments, and some of them look absolutely horrendous. I won't read it verbatim, but 25 guts of elixir vitriol doesn't sound too good to me, and neither does vinegar or a course of sea water. Fortunately for two of these good people, they did actually get an orange and a lemon. So we, of course, now know that that is an effective method of managing scurvy. For some strange reason, which of course we don't really know, Lind actually concluded that pure dry air was the best treatment, and he recommended fruit and vegetable as a secondary recommendation. Now this is a reasonable attempt at a clinical trial. I'll not look at my clinical trial colleagues in the front rows here. It's a reasonable attempt, but there are some things that Lind failed to do. And the thing that I will pick out is that he failed really to randomise the patients. So let's just have a little look at what randomisation is and what randomisation does. So in a modern clinical trial, you can choose to participate in a trial, and some of you in this room may have been asked to participate in a clinical trial. It is completely your free choice to do so. So we'll get that one out of the way and parked. What happens then is that the choice of treatment that you get within that clinical trial is removed both from yourself and from your doctor, and it is allocated by something that we call randomisation, which is the flicking of a coin, in essence. In fact, we use systems that are considerably more sophisticated than flicking a coin, but it is the equivalent of flicking a coin. So why do we do this? Well, it ensures that the individuals who are in the two comparison arms are pretty similar, as similar as we can possibly get them. And I've got this slide just really to show you, hopefully to demonstrate and make this principle a little bit easier to understand. So there's a slide here of purple, blue and turquoise patients who are going to enter a clinical trial of treatment A and treatment B. We think or have some evidence that the treatment A works best in purple people and treatment B works best in blue people. But actually, we want to estimate the effect of this treatment in all types of people. So if we want to get a really good, unbiased estimate of how well this treatment works, we have to ensure that there is a balance of blue, purple, and turquoise across the treatment arms, so the different arms of the experiment. If we flick the coin enough times, this is just simple probability. If we flick the coin and say heads, you go to treatment A, you have an operation. Tails, you go to treatment B and you don't have an operation. Eventually, if we flick the coin and allocate sufficient numbers of people, we will know that we've got equal balance of those blue, purple and turquoise people within those two treatment comparisons. Now, that's important because now all of those biases that we know about are equally distributed, and it sets up a fair comparison for that clinical trial. The really important thing that it also does is that the things that we don't know about the human body, and actually we don't know how much we don't know, but I'm going to tell you, I think we don't know quite a lot about the human body. Those things we don't know about are also allocated across the two groups evenly because we've used this system of tossing a coin. So for me, this is absolutely a fundamental for designing a good experiment. You have to go through this process. I think it's a little bit difficult sometimes for us to communicate this principle well. I'd be interested to get your kind of feedback on how easy you find to understand it, but it gets kind of a little misinterpreted. We do some work with young people, and the term random is not really what I think of as being random. It kind of implies a bizarre behavior of some description, and the lay kind of term is a little bit different, I think, than possibly the term that we think about. But we do try our very best to explain this, usually on an A4 sheet of paper, which is what we're required to do. And as you can see, it can get quite complicated. It's not quite a perfect science. Probability will not completely eradicate all chances of there being a difference between the two treatment arms, but it's as near perfect as we can get it. So let's go back in history. So we kind of did James Lind, and now we've come forward a good number of hundreds of years. This is a slide from the Second World War, I hope. Somebody in the audience, no doubt, will put me right if it's the First World War. But anyway, the very first clinical trials that were really randomized clinical trials that were run were actually trials of penicillin. And the army surgeons had started to use penicillin in the field, and there was an attempt to try to run a randomized controlled trial. And actually, this study, funded by the Medical Research Council, failed because the surgeons had already seen how beneficial penicillin was, and they did not want to randomize their patients. And this is a problem that we can encounter when we try to run clinical trials, and it's something called a lack of equipoise. So it means that either the clinician or the patient or the investigators are not in balance about how they feel about the treatment. And this can lead to all types of problems. So, equipoise and balance. The second trial, which the MRC funded, was a much greater success. That was a study of treatments of TB and actually paved the way for a whole new generation of methods of managing tuberculosis. So, I've talked a little bit about human flaws. I'm going to say that the environment in which we do clinical trials is somewhat supercharged. Not my environment, unfortunately. But if you look at the bigger picture, there's a lot at stake here. There's a lot of it at stake for pharmaceutical companies. There's a lot at stake for inventors of new treatments. And a whole heap of conscious and unconscious biases can seriously derail an attempt to run a randomized controlled trial. And this is why there's so much regulation in the field. And my life is about regulation and ensuring that we are adhering to the regulation. Understanding why the regulation that is there is really important. And I think it's hopefully a great reassurance to the public to understand about the regulation. So the first real flaw that I'll talk about is what we call subversion. So in the old days, we used to randomize patients, first of all, by an open list. So, a long time ago, we just basically could flick a coin or just generate a list of random numbers, and you could see which number was coming next, particularly on a list. And you could choose, if you knew which treatment was going to be administered to which group, you might choose to hold back a patient that you didn't think was going to do so well on that treatment. Or, you can just see how the best intentions could really upset an experiment. So it's a little bit like peeping through the keyhole. Then we moved on to have randomisation, allocation, concealed in an envelope, and we used to have numbered envelopes. But then, very fortunately, one of the great advantages of computing is that we've been able to move this allocation onto computerized systems. And now what we do is we register somebody into the trial. It's done through an independent randomization service. So we ring up somewhere and we say, hi, I've got Mrs. X. She would like to come into the trial. Please, can I give you her information? We check that she's eligible, and then we generate the randomization. They generate it via the computer, and then it's all registered so we can see what time the computer was accessed, what time the phone call was made, all of these types of things, and the allocation is given back. And that's really important because it makes that a much more robust process, and it's much less easy to tamper with. And I'm not implying, and I don't want anybody to go away, that every randomized controlled trial has people tampering with it or wanting to, but there is the risk of that happening. And I think these new, more modern methods of doing the randomization are really helpful and very important. So let's go on to the next one. Resentful demoralisation. And this is the first bias, the subversion, is usually a problem with the clinicians or the investigators. Sometimes it's a problem with patients, but not usually. Resentful demoralisation is usually a patient-led or participant-led bias. And what this is, it's basically voting with your feet. So if you go into a trial where you can see quite clearly what treatment you're having, and we find it difficult but not impossible to hide it from you. So for instance, surgery, if we say we're going to remove your left leg in one arm of the trial, and in the other arm of the trial, we are going to leave both legs intact, there's no hiding that. And quite often what happens, when people come into a clinical trial, they quite often have an idea about what treatment they think is better. And some people, naturally, this is just a natural part of being human. What's in it for me? I might get that treatment, or actually sometimes it's I might be able to have that diagnostic procedure that I can't have on the NHS. These are just human things. And if you don't get the treatment that you like, off you go. You say, I'm not actually staying in this clinical trial. I'm now a little bit hacked off with the allocation that I've got, and I'm going to do a walk. I'm not going to complete the questionnaires, and I'm not going to allow the researchers to track my information. What we then get is we get an imbalance again that's kind of just gets built into the experiment after we've done the randomization, because we can't retain people on one side of the trial. So we do lots of things to try and help with this. We invent placebo treatments, which are dummy treatments, and all patients, when they enter a clinical trial, are told if they have a chance of having a placebo treatment. Placebo treatments for medicines are reasonably easy, but not cheap to generate, so you get something that looks the same in terms of the packaging, hopefully tastes the same, kind of pops out the same way, so you really don't know whether you're getting the true medicine or not. It's over the last 5 years, and in particular in this hospital, we've been doing a lot of work trying to create placebo surgery. So surgery has a really, really big effect on people's minds. I mean, if you just think about it, the build up to a big operation, you've got yourself on the waiting list, you've kind of, you've made it through all of those hoops. It's a big deal to have an operation. and doing surgical trials is really quite challenging. So we have now managed to invent some placebo surgical studies. You will go down to theatre. It will appear as if you have had the operation, but actually you haven't had the active component of the operation if you're in the control group. So in my own field of orthopedics, we like to remove things. You know, we like to remove discs that are in knees, little bits of bone, that type of thing. And that's what we call the active part of the intervention. And what we do in these placebo-controlled studies is we actually give you an incision, but we don't do the active part of the operation. Now, I can see the lady, fourth row back, grimacing and thinking, oh my goodness me. And believe you me, actually, I think a little bit like you. I found this difficult, actually, to kind of think, why on earth would you do that? But I have to say, I mean, all people always know before they go into the trial, this is what could happen to you. So you make a free choice, and that is not hidden from anybody. And this is the gift that people give to us when they come into a clinical trial. They give this gift of saying, I don't mind which route I go down. And it truly is a gift, and I understand that completely, but it's not, you know, completely hidden from you, but it's not hidden from you at all. And that would be completely wrong to hide it from you. But it's that gift that you give to science and to medical understanding that's so important. We don't use them all the time. Interestingly, placebos are used sometimes in their pure form in medicine. But for somebody like myself, with my clinical background, I know that reassurance is really important to people, communicating with people, and just saying, oh, I'm really sorry, that looks like it's really sore. That's important, and that's a part of being a human. Sometimes that's what medicine is, and I personally don't think there's any wrong in that. I think our health service should provide that type of reassurance to people and that type of care. Sometimes we kind of like want to test that against an active treatment. So we won't kind of like, we use the placebo effect as a treatment as we would do in normal practice. I've probably made that a little confusing, but anyway. Okay, let's move on to another human flaw now. This actually I don't think is a human flaw, but this is a very, very common myth. The game of chances. Clinical trials are based on chance. So already we've talked about randomisation, where it's about the chance of me being allocated to surgery or no surgery. We then continue this theme throughout the clinical trials that we do, and we analyse the chance of having a good recovery using statistics. So the kind of chance thing is what it's all about. And of course, statistics tends to have a bad name. You hear this? Lies, lies, damn lies, and statistics. I'm going to actually say that I think that statistics is one of the most powerful tools that we have to understand the human body and to understand when we have effective treatments. But it's like anything. If it's put into the wrong hands, it doesn't do very well. That's just the same as if you put a sports car into the hands of a kind of, I'll just say into the hands of the wrong driver. We'll make no comment on that. Then that's also, you know, kind of recipe for disaster as well. So statistics really important. We always calculate a sample size for a clinical trial because we know that we have to have a certain number of people to kind of be able to see the differences and ensure that then we're not just seeing something by chance. So some of these trials are really very big because the thing we're looking for is not very common. And I'm going to show you an example of that. And that's kind of this idea that we're trying to look for sometimes a small difference against really quite a lot of background noise, a lot of kind of, the signal to noise isn't terribly high. And we take great care to make sure that we calculate the chances of seeing a correct outcome. We have a statistical analysis plan for all well-conducted trials, and what that is it's a clear specification of how we're going to analyse the data, and that's pre-approved by an independent data monitoring committee. So there's independent oversight of that process. Okay, let's move on to the law. We're going to talk a little bit about staying within the law. I've already said that this is a highly regulated area. Italian policeman, I wouldn't want to mess with him. This is just a little reminder of the kind of profit margin that is out there in medicines development. There is an 18% profit margin on pharmaceutical development. That's the largest profit margin. It's at the top of that kind of rack of profit margins for all industries. So it's very lucrative. It's also, it is lucrative. It requires a large investment of money as well. And the pharmaceutical companies in particular are always, kind of trying to ensure that they can kind of maximize the chance of finding a positive medicine, because they start many things. And I have to say this because if they were here, they'd go a little mad with me probably showing this, but they have to put a lot of money in to find a medicine. So there have been a whole series of declarations and various laws, and I've probably got the dates wrong here, but anyway, the Declaration of Helsinki is the most important one. And this is not actually a law. It's what it says on the packet. It's a declaration, and it's an internationally held declaration that has been updated many times. And this basically goes through every little detail of what you should do in the tricky situations of a clinical trial. And it provides the template by which law is then passed. And we have plenty of laws passed in this area. So, for instance, the Declaration of Helsinki says that you have to have a relevant control, and we can't just kind of say, oh well, it's no treatment. You have to ensure that you give participants of the clinical trial the best available normal treatment. So it's all of those types of things packaged up. The EU clinical trials regulation is absolutely enormous, but it is important. It's the bane of some of our lives, but nevertheless really important. We have the Data Protection Act. We have an extension called the Mental Capacity Act, because we have to protect very vulnerable people who can't give consent, either because they're unconscious or because they lack the cognitive ability to give consent. So there's very clear guidance in that area. The Federal Drugs Administration has its own requirement for medicines and I think now devices trials. European Medicines Agency, Medicines for Health Regulatory Agency in this country, and all of this translates into a very complex governance pathway, which we have to follow. And which I'm very willing to follow. It's actually executed through something called a local sponsor for a study. So the sponsor for most clinical trials around here is the University of Oxford, and it has a large clinical trials and research governance office that look to ensure that they're happy to what we call sponsor a trial through this system. So they're the first layer of scrutiny. Just depending on what type of study you've got going, you have to go through various other layers, but eventually you're allowed to take the study to the ethics committee. And the ethics committee and the health regulatory authority is really the place where the decisions are made. And for investigators like myself, I have to go to the ethics committee, I have to present myself, I have to present myself for questioning, and on occasion, the ethics committee can be a little bit of a hair-raising experience for us investigators. Kind of contains a very broad spectrum of people, lay people, scientists, all types of interesting people. So this is really the key things. The concern for the interests of subjects must always prevail over the interests of science and society. That's what the Declaration of Helsinki tells us. There must be independent oversight. We must, of course, have consent. We should always publish the results of a clinical trial. They give us guidance on what would be a reasonable comparator and placebo. And for those of you who might be interested in this, I found a very nice summary on Wikipedia. And I'll just put a slight disclaimer there and say, of course, that Wikipedia isn't always accurate, but for kind of getting a fast update on something, I think it's useful. And I think I found it helpful. So I'm going to just give you an example of a clinical trial. And this is a trial that I've been involved in. I just chose one of my own because it's easiest for me. I've been the chief investigator of many trials, and I've also led up teams of people who have done trials in other areas. And that cuts across trials in dementia, trials in falls prevention, trials in intensive care, trials of medicines, trials of surgery. My job these days really is to support people who have to do this very complex work and to keep them going and to keep them enthused about it, because it's a long, slow road. Some of these trials can take us 7 to 10 years to finish. And I should imagine Jane sitting on the front row. Longer? Yeah, you know, it's a long old haul. So let's look at an example from my own area, preventing falls, what works. So falls are really important. Cause of injury, death, institutionalization, dependence, depression. They're not great things to happen. Some people bounce up fine and they go and get on, but for some people, they really cause a problem, and they're particularly a problem for older people. So we don't actually know that much about what helps to prevent falls. There's some evidence to suggest that falls clinics might help, and there's some evidence to suggest that exercise helps. But these are kind of quite expensive interventions to deliver at scale if you consider that 30% of the population over 65 is at risk of falling. You know, that's actually quite a big ticket item for the government to invest in, and it really needs to know, actually, can you prevent falls through these different interventions? Is it any better than just providing people with advice? Does it prevent fractures? And how much does it cost? Because the government has to decide ultimately whether it invests money in X or Y. And we don't have that information. So we designed a randomised controlled trial. And what we do in this trial is that we randomise to 3 different interventions. Advice, so you just get a leaflet and some advice about how to manage your fall, and we hope that people are good citizens and that they manage their own health well. It's the kind of nirvana of healthcare, particularly as we kind of move into more constrained budgets. A structured exercise program is the next intervention that we're testing. And that is delivered by physiotherapists in your local physio department, and you have that over a 3 to 4 month period. And then the kind of superstar, the kind of gold star service, the multifactorial fall prevention clinic, where you come and you see the geriatrician, you see the nurse, you see the physiotherapist, you see the occupational therapist. And you might see them over 3 months, quite a few times. You might see the podiatrist, the optometrist, as they go through all of the different kind of parts of the body that contribute to falling. And as you can see, the healthcare bill for that begins to stack up quite substantially. So we did something a little bit different with this study. We actually randomised practices as opposed to individual people, and this is what we call a cluster randomised trial. The reason for that is that the risk of contamination of treatments was too high. So if we had gone into a single practice and said, nurse A, I want you to deliver this to this patient and that to that patient, the treatments are too similar. And the risk of nurse deciding, actually, that looked like a really good treatment, I think I'll just give this person a little bit of that, was too high. So we cluster randomised this. We decided that we needed to study 9,000 patients. That's a big trial. It's not as big as some of the ones that my colleagues here do. They do even bigger ones. But just to give you an idea of what that actually means, that means that we have been involved with 64 different general practices. We have to find those general practices and get them all on board. They're recruited from across the country so that we get a good generalizability from the sample. We want to do a really good piece of work to help inform government policy. We don't want a lopsided Oxford-only sample. I don't know if it would be good or bad if we had Oxford-only, but anyway, it would be something, this unique and special place. And we then have to think about, okay, what is it like? What are we going to do to the services in each of these areas, because we use the routine services to deliver the treatment? Are we going to overload them? So we have to think very carefully about how we sequence and manage all of this project. And at the same time, we have the Department of Health asking us, what are we spending every penny on, and why aren't we going quicker? There's quite a lot of pressure, as there should be. It's taxpayers' money. This study cost £2.2 million. So it is a lot of taxpayers' money, and we have to do a good job, and we have to do it as quickly as we can. How do we find these patients? Well, this is another of the great things with modern computing. Actually, what we do is we build software to interrogate the GP lists. In fact, the GPs do this so that the investigators, we can't just have your details. We have to go through an intermediary person. And we build a software platform that the GPs can use, and I've got some really great software engineers working with us, and basically they kind of rack through and they find us a sample, and then we mail out to people to ask them to participate in the clinical trial. And for this trial, the invitation to participate is entirely by post. So what we put in the letter and what we put in the patient information leaflet has to be not only legally correct, but it also has to hopefully attract people's attention and help them to understand. So there's a little bit of a balance. There's a bit of an art in writing these so that we don't get pulled up by the ethics committee and say, no, you're being coercive. But never, we don't want to put people off either. We want to be able to do this. So writing the letters and thinking about this process is a challenge. And then, of course, what happens is that we take from each GP practice 400 patients, randomly selected. And that means that we have to pre-prepare all the packs for 63 practices. They have to be posted from the GP practice. And then there's a whole series of stuff that goes on. But just to give you an idea of the letters that go out to one practice, what you can see in this corner is just the packs that we have just for one practice. And if we don't get a good enough response rate, we have to do it all over again. So it is quite a kind of mechanical grind. And the teams that we have working on this type of work are a combination of people with administrative and project management expertise, as well as people with the kind of experimental expertise, clinicians and patients as well. All of the data has to be checked. We have to have thought about, people love to ring us up actually, and that's really nice. But when it's 9,000 people, it's a little bit less nice. But we do like to hear from people, and people will kind of write notes on their clinical research forms that they return to us, their questionnaires, and sometimes actually they write some very sad things, and sometimes they write things that we have to respond to. So they will say, I feel that I can't carry on. Now, that's the difficult thing for us as researchers. We have to have a process in place to deal with that, and indeed we do. So it's just a lot of project planning and thinking about what's going to happen and being close enough to your experiment and the team of people who work on it to be able to pick up when something is not quite right that you hadn't anticipated. So that's really important as well. And that's Suzanne who goes through all of those questionnaires. We have indeed reached our target of 9,000 people. And if we hadn't have had this team of great people working on it, if it was left to me, this is what the Prefit Office, that's the name of the study, would end up looking like. So that's just really to give you an idea, the day of a life of a clinical trial. And this trial's taken us 7 years. and we're just about to start to analyse the data. So, I'm just going to conclude by saying that it's really useful to reflect when you see things on the television. And the thing that captures my eye is this Cancer Research UK, We'll Beat Cancer. We're 50% of the way there. Every one of those 50% is based on a clinical trial. It's based on a good quality randomised clinical trial. So without the randomised trial, that clear-cut information that we get back from trials just wouldn't be there. We would be losing our way because we couldn't have confidence in the experiments that we do. And there are just, I mean, almost all medical interventions, all medicines have been through clinical trials. There's some really, really important work that's been done through Oxford on things like aspirin, heart attacks, strokes, all done through randomised controlled trials, TB, vaccines. There are the greatest number of people who have been randomised into a bowl of vaccine trials is here, because people are running randomised controlled trials of these various different vaccines. So as you watch the television and you see all of this, just think about all of that medical advance. And I'm going to conclude by saying I do actually think that the clinical trial is the most important medical invention. I keep saying medical intervention, invention. And the reason is that it helps us to have confidence in all those inventions that we've been testing and trying to progress through to improving patient care. So that's the end of my presentation. I'm really happy to take any questions, or indeed just hear comments from people. I know sometimes people just want to make a comment and not a question.